In this video, I'm going to discuss how to become a pro at answering multiple choice questions. Often students tell me that they think that multiple choice questions are easy to answer until they take their first exam, they get their grades back, and they see that it didn't go well at all on the multiple choice questions section. Perhaps you were given very simple multiple choice questions in the past to answer in your biology class, the kind that it's very obvious to see which the correct answer is, and the options maybe not very applicable to the question, so it would be easier then to choose the answer by just glancing over them. But in biology college courses, the answer options are not so obvious. In fact, most of the students tell me that when they look at the answer options, it seems like every option could be a correct answer. So I'm going to give you some examples here on how to answer multiple choice questions. I'm going to go over my suggested method first, and then I'll end by telling you what are some of the other methods you could also try. I'm going to use this example. This is a multiple choice question, and it says, which statement is not true about covalent bonds? Now, you'll see that I did not list the answer options here. In fact, if you have a Scantron or an extra sheet of paper with you, I would suggest taking your Scantron and cover the answer options, A, B, C, D, or A, B, C, D, and E, so that you cannot see the options when you're reading the question. The reasoning behind that strategy is to keep your mind focused on the question. So often students will not read the question properly. They will not think about the question, but they'll just start glancing through or reading over those answer options, and too hastily then choose often an incorrect answer. So this is what I suggest you do. You close those options and you have just the question statement in front of you. Now let's see, what does this question ask us? Which statement is not true about covalent bonds? So now I would go ahead and I would make a couple notes here. Now, I know this depends on your exam or test paper that you're getting. Some instructors don't want you to make a single note or write not a single word on the exam paper. I'm not that way. I print out a copy for each student, and they can write on their copy as much as they want. In fact, I believe that that's the best way to answer questions and not trying to do everything in your head. But if your instructor says you're not supposed to make any scratches or notes on the paper because the instructor wants to reuse that paper copy, then perhaps you could just ask for a clean sheet of scratch paper that you can then do your notes on. But you'll see that I highlighted here which statement is not true about covalent bonds. So the question is asking about covalent bonds. So this is where I would start. I would make a couple notes here for myself, thinking through what I remembered and from my studying of covalent bonds. So probably you would think along this lines. I remember I studied about or learned about ionic bonds, and I learned about covalent bonds, and you may remember you learned about hydrogen bonds, and perhaps you also touched on van der Waals forces. So if you have that little list in front of you, maybe you can remember that covalent bonds are the strongest type of bond we have compared to ionic hydrogen and van der Waals forces. So going over these other kinds of bonds, you could now recall maybe an example in class. Maybe you learned an example of an ionic bond forms between sodium ions and chlorine ions, and it forms sodium chloride. So here we have the attraction between opposite charges of molecules, of ions, and those create a bond, a molecular bond that is an ionic bond. So we have an ion charge positive and an ion charge negative, and there's an attraction between them. So typically how this is explained in the classroom is that there is a giving away of an electron, and the other one is accepting the electron, and that's how that ionic bond is created. Then maybe you can remember from your class or reading in the textbooks, etc., that a covalent bond is where electrons are being shared, and that there's two kinds of covalent bonds. You can have a polar covalent bond and you can have a non-polar covalent bond. So if there's a polar bond, that refers to equal sharing, and if it's a non-polar, if there's a polar bond, then it refers to unequal sharing, and if it's non-polar, it's equal sharing. So for example, you could have, if it's an unequal sharing, you could have some electrons spend more time towards one side of the molecule, and then it creates on that side of the molecule a positive charge, and then on the other side it creates a negative charge, because here is where the electrons are spending more time on this end of the molecule. So this is what we call a polar scenario. Whereas if there's equal sharing, then the electrons are spending an equal amount of time on either side of the molecule, and the charge there is equaled out, so there's no charge across this molecule. Right, then we move on to hydrogen bonds. Hydrogen bonds, we learned in class, are the weakest type of bond, and those typically form between molecules where there is a hydrogen ion that is attracted to a negative charge. So this is often discussed in water as an example. There's many hydrogen bonds in water. You could also have some hydrogen bonds in DNA molecule, for example, between the nucleotide bases. So this is definitely our weakest bond because they're easily formed and easily broken. And lastly, we learned about the physical force that there is between some ions and atoms, and those are referred to as van der Waals forces. And this is more of a relationship or attraction that occurs because of proximity to another atom. So you see, I haven't even looked at the answer options. I made for myself a little summary here about the different kinds of bonds. Now look, if you don't have time to go through all of the kinds of bonds, then at least do it for covalent bonds, right? At least write down, I remember that's the strongest type. It's where electrons are shared. You can have polar, non-polar scenarios or develop. And so now only you go look at the answer options. And I do want to point out this. You are really trying to eliminate the incorrect options first. So you could scan over all of them, or you could do them one by one. So let's zoom in here, and we see our answer option says here, a covalent bonds form when an electron is completely lost or gained from an atom in option choice A. And so remember, we wrote down that it is shared, so it cannot be that one. Right off the bat, you can cross that out. It is not completely lost or gained, right? It is not what a covalent bond is described as. Okay, so B, a covalent molecule contains one or more covalent bonds. Well, a covalent molecule typically is going to have one or more covalent bonds present there. Let's see. A single covalent bond is drawn as a line between two atoms. If it's a single bond, it's one line. Yes, it's going to be a line. For example, you can have a covalent bond between a C and an H, a carbon and a hydrogen. D, a pair of electrons is shared between two atoms for each covalent bond. Yes, that's what we learned. A covalent bond is sharing of electrons. And then shared electrons, option E, allow an atom to complete its outer electron shell in that covalent molecule. Oh, we didn't talk about the outer electrons, but that typically is what all bonding is striving towards, right? to complete that outer electron shell. Here they do again refer to shared electrons, so that is spot on. So let's go back here. The question was, the statement says there, which statement is not true, right? And we have chosen here from our options here that covalent bonds A form when electron is completely lost. Again, this is not true. So this one will be the answer that we select. And so we will bubble on our Scantron or circle question A as the answer option. So this was a process of going through all of the options and checking which ones are correct and which one is not correct. Let me give you another example of a question that also can be helpful when you are drawing a little diagram to try and solve the problem. This question reads, cells of the pancreas will incorporate radioactively labeled amino acids into proteins, and this tagging of newly synthesized proteins enables a researcher to track their location. In this case, we are tracking an enzyme secreted by pancreatic cells, and the question asks you, what is its most likely pathway? Now, this question is a question about some cellular biology. So typically in a chapter in your biology curriculum, you learn about cells, you learn about the organelles within those cells and their functions. And you should have learned about enzymes also, but it gives you a clue here as to what an enzyme is. Even in the question, it tells you that amino acids are incorporated and built into proteins. And that should remind you that, hey, I learned in class that an enzyme is most of the time, in fact, a protein. Not all enzymes are proteins, but most of them are. So they're asking you, what is the most likely pathway here to produce a protein? What is the most likely pathway to produce a protein? And an example is an enzyme in this question. You know, they are including an organ here, the pancreas. Well, it doesn't really matter which organ they're referring to. In a cell, you're going to have an endomembrane pathway. So let's draw a cell. This could be a pancreatic cell. Typically, you have your nucleus in there with your nucleolus. And then in that cell, you're going to have some rough endoplasmic reticulum associated with the nucleus. Of course, this is your endoplasmic reticulum that has the studs of ribosomes on it, but you'll also have some smooth endoplasmic reticulum, sort of connected also to this endomembrane system, which the nuclear membrane is a part of. And on the smooth endoplasmic reticulum, there's no ribosomes. And then from what you've studied about cell biology, you have learned that there's also a stack of flattened membranes referred to as the Golgi apparatus, and typically they're sort of drawn like a stack of pancakes almost in the cell. And you learn that there are some vesicles that travel between both the smooth and the rough ER and the Golgi complex or apparatus, and then also some vesicles that bud off and move towards the membrane, the cell membrane of this cell. So there can be movement here from the smooth ER and to up to that point in the cytoplasm. You can have then movement all the way through and reaching an area in the Golgi called the cis phase of the Golgi, and then at the trans phase you're going to have some vesicles leave again, and of course these vesicles can fuse with the membrane of the cell and then exit out of the cell. So thinking on how these proteins are produced, of course, this question doesn't really ask you about transcription and translation there in the nucleus. It's more so focused on the secretion by the pancreatic cells. So I'll also underline that. So you kind of have a pathway drawn in here. Maybe you can just label a couple things here for yourself so that it stands out. This was your smooth endoplasmic reticulum. This was your rough endoplasmic reticulum. Over here you had your Golgi, and there is your cell membrane. you also had some of these vesicles in here, and maybe some of those vesicles do have digestive enzymes in them, and then they are referred to as lysosomes. Okay, so that's a quick little summary of the membrane system in a cell. So if you want to jot down so that if you come back later to this question, you can see, oh, this is what I drew here. I drew a quick little illustration of the endomembrane system. Now, let's go look at what is the most likely pathway of an enzyme secreted by the pancreatic cell. And we look at our answer options. All right. So number A, answer option A, says we go from the endoplasmic reticulum to the Golgi and then to the nucleus. And right off the bat, you should be able to eliminate that one and say, no, that is not possible. We do not have it moving to the nucleus. In fact, the instructions to make the proteins originate here in the nucleus from the DNA, of course. So we're not going to go back to the nucleus. We want to secrete that enzyme that we're producing. So the instructions are in the DNA, but it's going to be used to make that enzyme to leave the cell. All right, so it can't be A. How about from the Golgi to the ER to a lysosome? Well, no. When we discussed our little diagram here of the endomembrane system, we said that at some point the Golgi apparatus is going to play a role, but that's not the first step. In fact, the ER, either the rough or the smooth ER, will be a step before that. Then let's look at C, nucleus ER Golgi. Well, maybe that's an answer option, right? So maybe you can circle that for yourself because that certainly sounds better. We start in the nucleus, we go to the ER, and then next we're going to see the Golgi organelle after the endoplasmic reticulum. But there's two more answer options, so let's quickly look at those. It says D, ER Golgi, vesicles that fuse with the plasma membrane. So let's see. This includes some information about the vesicles fusing with the plasma membrane, and this we didn't see before, and that's important because that tells us that it is being secreted, right? If it's going to fuse with the plasma membrane, it's going to go out of the cell. Secreted means to be released out of the cell. So maybe we do have this as a possible answer option, right? So we could circle that one. Maybe it is this one. How about E? The ER, then there's an arrow to lysosomes, and then there's an arrow to vesicles that fuse with the plasma membrane. So basically the difference between D and E here is in what we see the lysosome here is given first, but here the Golgi and D is given first. Now we know that the ER can release lysosomes, but those lysosomes will have to go to the Golgi to be processed and packaged. So there is a missing step here. You would have to have the Golgi involved before the vesicles can fuse with the plasma membrane. Those vesicles will be released from the Golgi after they were processed. So we can eliminate D. And then from C we can eliminate E. And from answer option C and D go back, now we can see that D clearly was the one that best described for us that there is a secretion, right? The vesicles that fuse with the plasma membrane and the Golgi was involved and the endoplasmic reticulum was involved. So our answer option here for this question is answer option D. Let me highlight that in a different color so that stands out. So you see here we had to approach it in a little bit of a different way. There was some that may seemed right, and then but we had to eliminate them or just really choose between C and D. So we eliminated 3 options, and it's always the best idea if you can do some elimination. And then really if you are still unsure about which of those 2 answers are the best option, I would, guess, never leave an answer blank, because you now have a 50% chance of guessing the answer correct, whereas before you didn't have such high probability of guessing it correctly. Okay, let's do one more example before I give you just another kind of strategy you may also think to use. Let's read this one together. It says here, a cell whose cytoplasm has a concentration of 0.02 molar glucose is placed in a test tube of water. So I would go along and just draw it, right? You're putting the cell in a test tube with water, and it says that the cell has a 0.02 molar glucose concentration in there. It is in that water containing also 0.02 molar glucose. Right, assuming that glucose is not actively transported into the cell, so there's no additional ATP energy involved here, which of the following terms describes the tonicity of the external solution relative to the cytoplasm of the cell? Oh, so they're asking us to specify here about the tonicity of the external solution, right? Our external solution is what is here surrounding our cell. So that's what they're asking us about. Sometimes it's helpful if you have a highlighter or two close by to make a little diagram like this as you read the question. Okay, so in class, from what you have learned, I hope that you've learned about these terms. A solution can be isotonic, a solution can be hypotonic, or a solution can be hypertonic. And we look at those prefixes in the word there to help us understand what those mean. Iso refers to same, hypo refers to a lower, and hyper refers to high. And the tonicity here is in the combining form of that term, right? So which solution here has the highest tonicity? Well, you should have seen right from the start that they're giving you exactly the same molar concentrations. There's a cell with 0.02 rather, and then the test tube has also 0.02. So this is an isotonic scenario. Helpful here was that you drew the diagram right from the start and you were looking at what is the question that they're asking me. And now just make sure one more thing. They're asking you to focus on relative to the cytoplasm of the cell, right? Did I compare the outside solution to what is in the cell? Yes, I did that when I was looking at these two molar concentration. They are the same. So my answer option here, you didn't look at them before, you just circle, it has to be isotonic. It cannot be any of those other options. So this is an example of where you would not even be eliminating the answer options. Of course, you want to just quickly glance over them and read them and to figure out if there's anything there that may be a little bit tricky, giving additional information. But from our little summary, right off the bat, we could summarize that it is definitely isotonic. So don't double guess yourself, especially if you know the work. Go with your gut answer. I've often seen students change answers and then they change it to the incorrect answer. Okay, now lastly, I do want to give you a little bit of advice here. Maybe I'll just zoom in here and make a few notes for you over here. Multiple choice questions can really make or break your exam grade. You could have a great free response section, but if you fail the multiple choice section, you can end with a poor grade for an exam. So some strategies you should remember for shorter type multiple choice questions. Think about the time that you're spending on a question. You know, there's this rule that you really should use 1 minute per question. But you can see as I was explaining some of these, some of them do take longer. So I don't think that that is a rule that is sort of set in stone. And moving too fast through a question obviously can hurt your answers as well. So maybe if there's some that are easier than others, it's maybe just a plain recall type answer for a multiple choice, then sure, use the time strategy. But I think it would be better to allow for you a block of time. So let's say you have 50 multiple choice questions and you have, say you have 25 points in the free response section. And let's say you're given 1 hour and 20 minutes to answer your whole, exam, to complete the whole exam. I would bargain to spend at least 60 minutes on the multiple choice section. But the best strategy I've noticed for many, many students, in fact, is to start with a free response. Start there, spend your 20 to 25 minutes there, and if you are not done, then pause there and go to the multiple choice questions and then do them. And you can much faster read through a multiple choice question than you can construct a free response answer. So in the end, you may do better by just the simple strategy of starting with a free response. The other option that I do want to mention, I would call this one option number 2. The technique I explained to you with using those two or three examples was not reading the answer options. But sometimes some students tell me that they have success if they read the answer options first, and then try to see from the answer options if there is a statement in there that seems contradictory. The one is the opposite to the other, for example, then maybe it would be that you have to choose between those two options. So certainly if you're struggling with using the first option, then maybe try option 2 for some of the questions. But as I said, I really like the first option best. And then another strategy you could also try is the sequence in which you answer your multiple choice questions. If your instructor did point out to you from which chapter the multiple choice questions came in your textbook, then yes, go ahead and do the chapters that you are more familiar with. So maybe, per chapter answering would be a good strategy for you. Some instructors don't do that. They just give you 50 multiple choice questions, for example. You may want to try doing number 1 to 50 and see how that works for you. I've seen some students do number 50 and they work backwards. They do 50 and then they work towards the start of the exam paper. That's up to you. I don't think that the first question on a multiple choice paper is necessarily the easiest question. Some students get the first question wrong and then it just goes downwards from there. So maybe you want to try breaking it down into sections and then decide, which section you want to spend more time on. If you're more familiar with enzymes, answer all the enzyme questions first, for example. So I'll make a follow-up video on this. If you post me some questions and if you give me some examples of multiple choice questions, go ahead and share them there in the comments and I'll help to explain how to answer those. I hope that you find this helpful and I hope that answering multiple choice questions will become easier for you and that you will become a pro at answering multiple choice questions.